Welcome, my dear friends. Regarding of our CPGET 2025 free online coaching, today's our resource person, Dr. Gogikar Bhagirath Sargaru, is waiting for us, my dear friends. So, sir, please welcome. Start your session. Share your screen, sir. Please welcome.
Thank you, sir. And, welcome to online participants. I think my screen is visible.
Yes, sir. Please click on slideshow.
Okay. Yeah. Is it clear? I think.
If possible, please click on slideshow.
I already done.
అయితే మీరు క్లిక్ స్లైడ్ షో నుంచి బ్యాక్ రండి, ఎందుకంటే ఇక్కడ అది కాలేదు, పెద్దగా కాలేదు.
Okay, it's now okay.
ఓకే.
Yeah. So, as per the CPGET PG entrance in botany subject, we go through the syllabus. After completion of the mutation part, we can. Today, our topic is genetic code and its properties, and as well as it covers the role of Gamow scientist in the discovery of genetic code and the Hubble hypothesis, and also some mutations based on the genetic code. Also, today we will discuss one by one. In most of the organisms, DNA is main genetic material, except in some plant viruses. But this DNA commonly we are referred as blueprint of the life, or sometimes it is a genetic blueprint, because it contains the genetic instructions or genetic information used in the development and function of the all living organisms. So, what is the main role of DNA in the living organism? First one is it store the genetic information, and as well as it transmitting the genetic information, and third one is guiding the cellular processes. That means DNA has many nucleotides in its molecular structure. It is code whatever the nucleotides arranged sequentially in the DNA molecule, which provides instructions for different cellular processes like growth and also development and different metabolisms which are performed by cell organelles. So we can say simply genetic code is nothing but it is a dictionary that corresponds with a sequence of nucleotides and sequence of amino acid. That means a special kind of relationship between the sequence of nucleotides and a sequence of amino acids. How a protein molecule synthesized in the living cell, which part of the DNA having that source or the information in the form of genes. So the genes are expressed and it reveals the information and passed to a intermediate molecule. With the help of intermediate molecule, it constructs the protein with the arrangement of amino acids in a sequential manner and linked with peptide bonds, and finally it modifications are takes place in the protein. Then it is released as a correct protein or an enzyme molecule in the cell. So genetic code is nothing but it is a set of rules by which whatever the rules are framed in the genetic code, by such type of instructions are information is encoded in the genetic material, that is either DNA or RNA, is translated into the proteins by living cells. So genetic code is nothing but a special kind of relationship between the information coded in the genetic material. Sometimes main genetic material is DNA, but DNA indirectly it gives the information to RNA. The RNA or DNA sequence of nucleotides is translated into the proteins by living cells. First time this genetic code term coined by George Gamow. So is also very important in the entrance point of view. In the discovery of genetic code, the two famous personalities, because after the discovery of DNA molecular structure by Watson and Crick, both the scientists, then we can understand how proteins are encoded begin after the structure of DNA and how a protein molecule is constructed in the cell. First of all, we understand the DNA molecular structure, which is revealed by Watson and Crick. So later one, George Gamow postulated that a three-letter code must be employed to encode the 20 amino acids, standard amino acids used by living cells to build the proteins. According to Gamow, he said that three-letter code, that means a sequence of three nucleotides must be operate to identify a particular amino acid in the construction of a protein molecule with the help of ribosome cell organelles in the living cells. So, how this genetic code is discovered? First of all, in the DNA molecule, we know very well four kinds of nitrogen bases are there. That is adenine, guanine, thymine, and cytosine. So, which are combined with the respective sugar and as well as phosphate group, it converts into GTP, CTP, TTPs, ATP like that. They are organized. These four letters are organized into codon. So how the codon is perfectly matching to the amino acid number. In a natural cell, the protoplasm or the cytoplasm containing 20 standard amino acids, but recently one or two discovered, which is also written in some books. For 20 amino acids, how they are identified by the genetic instruction provided by DNA molecule. So, for example, in the DNA molecule, four type of nucleotides are there. So that is nothing but one is A, AMP, GMP, TMP, and CMP. Cytosolic acid, thymidilic acid, guanolic acid, adenilic acid, like that. So these four nucleotides individually recognize amino acid. We can assume it. So each codon should have three nucleotides to impart the specificity of each of the amino acid for a specific codon. How a amino acid recognized or identified during the protein synthesis. So if a single amino acid is determined by a single nucleotide, that means one nucleotide only. So it gives only four combinations, that is AMP, GMP, TMP, and CMP, like that. So but these four combinations are not enough to identify the 20 amino acids. So in the living cell, we have 20 amino acids are there, standard amino acids. If four combinations are not sufficient to identify the whole amino acids. So that's why they assume two nucleotides combinedly determine or recognize the amino acids. So if two nucleotides, how many combinations we are expecting? 4 into 4. 4 into 4. That is 16 combinations are we are expecting. Both the nucleotides are combinedly helps in the identification process. So 16 combinations also not enough, not adequate to identify the 20 amino acids. That's why they assume maybe 3 nucleotide sequence. That means the triplet, 3 group of nucleotides is directly helps in the identification of amino acid. So in this case, we can write 4 cube. What is that? 4 cube. 4 cube means 4 into 4 into 4. That is how many combinations established? 64 combinations are established. This is enough and also more to identify our 20 amino acids. So based on this, the discovery of genetic code takes place. The collection of codons is called as genetics code. How many how many nucleotide bases collectively represent a single amino acid? That is, 3 nucleotides are collectively represent a single amino acid. In this way, 64 combinations are framed. Okay, next slide. So the genetic code is a triplet codon. Triplet means 3 nitrogen bases or 3 nucleotides which are arranged sequentially on the DNA molecule. For example, ATG is a codon. This helps in the identification of a single amino acid. Another codon, CTA, GGC. In this way, group of 3 nucleotides commonly called as triplet, that is initiate the genetic code. As per the genetic code, the information pass to the protein molecule. The information helps in the construction of a protein molecule. So in this way, the DNA pass the information to the RNA. From the RNA molecule, protein is constructed in the cytoplasm with the help of ribosome. So this is in the nucleus, the DNA, it consisting of several genes. The genetic information passed through the dictionary of codons. So it is not directly involved. So that's why, as per the central dogma of molecular biology, the flow of genetic information from DNA to RNA, RNA to protein, and finally we are achieve a protein molecule either in the 3D structure or 4D structure. So this is the genetic codon dictionary circle with the help of 3 circles: inner circle, middle circle, and the third ring of third circle. It represent the particular amino acid. It is another type of pictorial representation of the codons. So how this codon dictionary established? Codon is nothing but triplet codon. You already just now we said. So it is a group of 3 nucleotides. In the codon, the first letter is nothing but first base. The second letter, that is the middle base of the codon, and the last letter, that is the third base in the codon. We can write like this: first base U, second base U, and third base U. That becomes triple U. Triple U is represent phenylalanine at the time of protein synthesis. In this way, total each block in the table, 4 amino acids, 4 codons are there. So 4 into 4 and 4. So total 64 codons are seen in this table. That is nothing but codon dictionary. So next, genetic code is a dictionary consisting genetic words called as codons. Each codon consisting 3 nitrogen bases, that is nothing but triplet. So in the previous slide, we can explain total 64 codons are there in the codon dictionary. Out of the 64, 61 codons are sensible codons, which are helps in the identification of amino acids, 20 amino acids. And 61 sensible codons represent 20 amino acids found in different protein molecules. 3 codons do not code for any amino acid. 3 codons do not. 3 codons means what? 3 is one codon consisting 3 nitrogen bases, but never 3 codons do not code for any single amino acid. Next. So out of the 64 codons, 61 are sense codons. 61 are sense codon. 61 is sensible codons. That means it recognize amino acid during the protein synthesis process. Few codons work as signal codons. They function as signal codons. Signal codons are two types: start codons, stop codons. So start codons is nothing but the codon which initiate the protein synthesis process by giving a signal. Those codons that code for signal during protein synthesis are called as signal codons. You know very well that is a starting codon, initiating codon we can call as దీన్ని మనం ఫస్ట్ codon అని initiation codon అని చెప్తాం, AUG. So this is the initiation codon in most of the organisms. Another type of signal during the protein synthesis are called signal codon. They code for amino acid, different amino acids. And two type of signal codons I am already told, that is terminating codon and initiating codon. What are the terminating codon and initial codon? Just we can these three codons. So these three codons called as terminating codons or nonsense codons, because it may not indicate any type of amino acids during the protein synthesis. That's why there is no sense and meaningless. It may not represent any kind of amino acid, any type of amino acid. So in previously it is called as amber, ochre and opal. These three terms are used for the identification of three codons: UAA, UAG, UGA, are nothing but is called terminating codons. AUG is a signal codon as well as a initiating codon. It codes for first amino acid in all proteins. In eukaryotes, it indicates which amino acid? Methionine amino acid. In prokaryotes, it indicates formyl methionine. So AUG is the initiation codon. It codes for first amino acid in all proteins. At the starting point in the mRNA, this codon recognizes methionine amino acid in all type of proteins. So this is the amino acid list, and representative letters generally we are used in the amino acid name with represented with 3-letter abbreviation: alanine ALA, arginine ARG, like that. So all the students definitely remember this short abbreviation forms also in the amino acid identification. So what is the difference between anticodon and codon? Codon is a functional triplet which are present in the mRNA molecule, but it is a reverse thing. Generally, anticodon we can see in the tRNA structure. In the tRNA structure, the anticodon represented by 3 letters, same like codon. But during the translation process, the tRNA anticodon pairs with codon of mRNA. This is the codon which is in the mRNA molecule, and this is the tRNA structure. This is the anticodon loop. It is also represented by 3 letters. So temporarily anticodon binded with codon of mRNA. So in the translation process, so the anticodon is which is present in the only tRNA. Next. Difference between codon and anticodon. Codon could be present in both DNA and RNA also. DNA we can generally call as sequence of nucleotides. In case of RNA, sequence of codons. But anticodon is always present in RNA, that is tRNA, never in DNA. We can't see in the DNA. And one of the important point, codons always written in 5' to 3' direction. And always the reading frame of the codon is in which direction? 5' to 3'. So, but the codon in the same way, anticodon direction is like this, 3 to 5 direction. 3 to 5, 3' to 5 direction, that is anticodon. 5' to 3' direction, that is codon direction. You always remember the DNA nucleotide sequence at the time of replication, transcription, translation, it moves from 5' to 3' only. Except in anticodon, the reading frame is always in 3' to 5' direction. So that is a reverse completely. Anticodon of some tRNA molecules have to pair with more than one codon. Later we will discuss about that one. Okay. Next. So what are the properties of the genetic code? Genetic code, it having a set of rules and which are framed for passing the DNA molecule into the proteins by living cells. So how the characteristics or the properties of the genetic code we can explain one by one. Genetic code is a triplet code. Genetic code is commaless. Genetic code non-overlapping. Genetic code degenerate. Genetic code is universal. Genetic code is non-ambiguous. And remaining points indicate the chain initiation and chain termination and reading frame of the RNA. So first we can go for triplet codon. We already learned a group of 3 nucleotides constitute as a codon. This codon represent one amino acid. This codon represent one amino acid. So in this way, 61 codons are represent 20 amino acids. So when experiments were performed to crack the genetic code, it was found to be a code that was a triplet. Genetic code is nothing but it is a triplet that we can call as codon. The 3 letters codes for nucleotides. What are the 3 letters? Triple A, triple C, triple U, AUG, like that, etc., etc., which are called as codons. We already seen in the codon dictionary. Once again, we can see like that. The genetic code is triplet. There are 64 codons are there. 61 is the sensible codon, 3 are nonsense codons. See here. So by the combination of first, second, and third letters, we are getting 64 codons. So first, one of the important property of the genetic code, the code is degeneracy or redundancy. That means more than one codon may specify the same amino acid. One of the amino acid which is coded by more than one codon. This is also called as degeneracy of the code. I'm already told that one. So 61 codons are there, but the standard amino acid number is 20. So 20 amino acids are identified by 61 codons. That means some amino acids among 20 which are identified by more than one codon. But a single codon always recognize a particular amino acid. A single codon always represent a particular amino acid. But a single amino acid can identify it with more than one codon. That these two statements are illustrate the property of degeneracy or the redundancy of genetic code. For example, except tryptophan and methionine, these two amino acids having only one codon, which are identified by only one codon. Remaining few amino acids are identified by sometimes two codons, sometimes three codons. So the 9 amino acids, namely phenylalanine, tyrosine, histidine, glutamine, asparagine, lysine, aspartic acid, glutamic acid, cysteine, have two codons. These amino acids are identified by two codons. Isoleucine has three codons. So this property called as degeneracy. Okay. Next. So degeneracy, this table illustrates that how many codons which represent a single amino acid. Arginine 6, leucine 6, serine 6, glycine 4, valine 4, like that. Isoleucine 3, and methionine tryptophan itself only 1. So how it is possible? So due to the variation in the third position of the letter only. So in these 4 codons, first 2 letters are same. These are the standard to identify the amino acid. In the third position, whatever the letter is present, it identify the same amino acid. That is, one amino acid may be specified by more than one codon. These third position we can call as Hubble position. Next, genetic code is non-overlapping. That means one nitrogen base cannot participate in the formation of more than one codon. One nitrogen base cannot participate in the formation of more than one codon. That indicates code is non-overlapping. For example, on the DNA molecule, these two codons are there. These two codons are there. CAT, like that. If it is non-overlapped, we are getting two amino acids only in the protein synthesis. These two codons represent two amino acids. If it is overlapped like this, it represents 4 amino acids. 4 amino acids. CAT1, ATG second one, TGA third one, GAT fourth one. But universally, genetic code is non-overlapping. That property indicates non-overlapping. So never a single nitrogen base participation in the identification of more than 2 amino acids. That is a thing you can observe by this picture. So next one is?
Sir, one doubt, sir.
Tell me.
Sir, overlapping code is not possible in protein synthesis, sir.
Overlapping?
Pattern is not possible.
No, not possible. Overlapping is not possible.
Okay, clear.
Okay. Genetic code is commaless. So in the DNA molecule, all the nucleotides arranged in a sequential manner, and there is no any kind of space or any gap to identify the particular codon position. the genetic code is commaless. Commaless means, which means that no codon is reserved for punctuations. It means that after one amino acid is coded, the second amino acid will be automatically coded by the next 3 letters. అంటే ఈ రెండింటిని వేరు చేయడానికి మధ్య మనకు ఎలాంటి చిహ్నాలు, సింబల్స్ కానీ, స్ట్రక్చర్స్ కానీ, స్పేస్ కానీ అలాంటివి ఏమీ ఉండదు. దట్స్ వై ఇట్ ఈస్ కాల్డ్ as commaless. అంటే for example, a sentence with 3 words consisting 10 letters. If you write the 10 letters separately, and we can also read the sentence like manner, but we unable to understand clearly because every letter is separated with a small gap. So in this way, just you can observe on the DNA molecule, nucleotides or codons are arranged in a sequential correct order. There is no any kind of punctuation marks to identify where is the correct codon and first codon, second codon, like that. So all the letters are arranged sequentially, and first 3 letters indicate one amino acid, automatically next followed 3 letters indicate second amino acids. That means there is no just you can read this quote. The genetic codon should be read continuously without spacing or overlapping. So it never overlapped, and it may not identified with the spaces. All the letters are clearly distributed with a specific manner. So 2 forms of genetic code without comma, genetic code with comma. So just you can see these 2 figures. So this is the non-overlapping comma free, non-overlapping with comma. So if it is overlapped, there is no chance like that. If it is non-overlapping and comma free, that is the first picture according to the genetic code is applicable. Next, genetic code is universal. So that means whatever the codons in the dictionary which having universal law, that means AUG is the codon for methionine. Sorry, that not the correct statement. Genetic code is the universal, but it is not strictly true. It is nearer to true, because in some cases one of the codon using for to stop the protein synthesis, that codon maybe indicates as amino acid in another location. For example, in the mitochondria, AUG codes for what? Isoleucine. AUG is codon for methionine in mitochondria. The same codon codes in the cytoplasm as isoleucine. And some little bit error is there, but overall it is nearer to the true, universal, near to universal, not strictly true. It is sometimes in the cytoplasm containing mitochondria and chloroplast DNA and mitochondrial DNA are exception for this rule. Okay, exception for this rule. In all living organism, genetic code is the same. అంటే AUG అంటే మిథియోనైనా వస్తుంది, ఏ యానిమల్లోనైనా, ఏ ప్లాంట్లోనైనా. దట్స్ వై జెనెటిక్ కోడ్ is universally acceptable. All living things share a common evolutionary heritage. So this tree-like diagram indicates that in most of the organisms, the AGA and AGG code for arginine in cytoplasm. But in mitochondria, they are confined as terminating codons. Exception is there for the mitochondria and the chloroplast. So this is the thing as per the genetic code property. Is it clear?
సరే, ఇది లైన్ అండర్లైన్ చేసి ఉంది సార్.
This one?
కాదు సార్, దాన్ని సెంటెన్స్ ని కొట్టేసినట్టు రాసిరు. మరి
అదే స్లైడ్ లో వచ్చింది, స్లైడ్ లో స్లైడ్ ప్రింటింగ్ స్లైడ్ అట్లా వచ్చింది. ఓకే. So next slide, we can go for the code has polarity. The genetic code has polarity. That is, the code is always reading in a fixed direction. So during the transcription, translation, our reading frame always in 5' to 3 direction. This is you can remember for this point. Genetic code always read in 5' to 3' direction. The code has a definite direction for reading of message, which is referred to as polarity. Polarity అంటే ధ్రువత, అంటే ఎటువైపు నుంచి చదవడం, ఎటువైపు నుంచి మెసేజ్ అనేది ఇన్ఫర్మేషన్ అనేది వెళ్తుంది అంటే 5' to 3' డైరెక్షన్లో. Reading of message from left to right, right to left will specify for different amino acid. For example, in the reading frame, we can read from left to right. It is what codon UUG. If it is read right to left, what type of codon we are expecting? GUU. Are you getting my point? The reading frame of the genetic code is always in 5' to 3'. That means left to right. So UUG, according to it, identify leucine. If the reading frame in reverse direction, that indicates which amino acid? Which amino acid? Valine. And that means it different amino acid will be incorporated during the protein construction, but not like that. The code has inequipolarity. The polarity in which direction? 5' to 3'. Is it clear? So chain initiation codons, you know very well, that is UAG, UAA, UGA. These codons never identify a single amino acid or not specify any kind of amino acid. That's why it is called nonsense codon. It helps in the terminate the protein synthesis process, to stop the protein synthesis process. So these codons are called with different names also, amber, ochre and opal. So these codons are not read by any tRNA molecule via their anticodons, but are read by some specific proteins called release factors. At the time of translation, these RF1, RF2, RF3 factors in prokaryotes and RF in eukaryotes, which act as a signal to stop the protein synthesis by reading the mRNA frame. The codon may be any one of these three codons, that may be UAG, UAA, UGA. So chain initiation, we already known that is AUG is the initiation codon. Next, what is meant by WGL hypothesis? How it explain the genetic code degeneracy? WGL hypothesis, according to the WGL hypothesis, which postulated by F.H.C. Crick, according to its hypothesis, in the tRNA anticodon, the third position is have a flexibility to can able to pair with one or two nitrogen bases, more than two nitrogen bases. So that's why in the codon, first two letters are very important in the recognition of amino acid. In the codon, third base, it can bind with anticodon of first base. Anticodon of first base. Anticodon lo first base, codon lo third base. So for example, this is a codon CCU. Okay. So this is the tRNA anticodon. Okay. So in the anticodon, it is the first base, second base, third base. Codon is always 5' to 3'. This is 3' to 5'. It is 3, 5. Okay. This is 5. This is 3'. So according to the WGL hypothesis, these first two letters are very important in the recognition of amino acid. The third letter is not at all having the much importance. It have the flexibility. How it is flexibility? In the tRNA anticodon, the first nitrogen base, it can able to pair with more than two type of nitrogen bases or more than two nitrogen bases. So this is mRNA codon, tRNA anticodon. So this is third position that is UBL position, it is called as. So according to in the codon, we can call as it is UBL position. Are you followed?
Yes, sir.
Yeah. Next. Wow. So in the tRNA anticodon, A letter is there. In the mRNA, it generally bind with U. C is there, it bind with G. So, but in the tRNA, anticodon position, anticodon represented by the first letter G, it can able to pair with C or U. In the same way, U is there, it can able to pair with A or G. So UBL base, that is called as. Sometimes in the tRNA anticodon, one of the modified nitrogen base is also present, that is called inosine. It can pair with adenine, cytosine, uracil, any one of the three. Either it can pair with adenine or cytosine or uracil. So that's why there is no importance in the third base of the codon to match with the anticodon at the time of translation process. So these are the UBL rules. So indicates how it is represented with another letter also. It receives the same amino acid. You can explain with the next slide easily. You can understood. So according to the Crick, to explain the degeneracy of the genetic code. According to this hypothesis, only the first two bases of the codon have a precise pairing with the bases of the anticodon, while the third one may UBL. UBL means it is not specific. For example, this is tRNA carry the leucine. This is also tRNA carry the leucine. So, but in the mRNA, what is the codon here? CUC. And in the next codon, CUU. But the tRNA bind with the CUC and the same tRNA carry the leucine, same amino acid, but anticodon is GAG. That means the third base G, it can pair with C as well as it can pair with U. That's why a single amino acid encoded with more than one codon. Are you getting my point? ఒక అమైనో ఆమ్లము ఒకటి కంటే ఎక్కువ కోడాన్లచే గుర్తించబడుతుంది. ఎందుకు గుర్తించబడుతుంది అంటే దానికి సరైన ఆన్సర్ UBL హైపోథసిస్ ద్వారా చెప్పొచ్చు. We can explain about the correct analysis for the degeneracy of the codon. The third base in the codon, first base of the anticodon, the pairing formula, the pairing match is different when compared to the first two nitrogen bases. It is fixed. But in the third position of the nitrogen base in the anticodon, it can pair with two or more than two nitrogen bases. That's why same amino acid carried by the tRNA. What is the main function of the tRNA at the time of translation? To carry to transfer the amino acid from cytoplasm to ribosome. కాబట్టి ఇక్కడ యాంటీకోడాన్లో మార్పు ఉన్నా కూడా మనకు సేమ్ అమైనో యాసిడ్ వచ్చింది. కారణం ఏంటంటే UBL హైపోథసిస్. దిస్ ఇస్ ద UBL బేస్డ్ పొజిషన్. ఇస్ ఇట్ క్లియర్?
Yes, sir.
This is the UBL rules. So G can pair with I means it is modified inosine. It can pair with U or C or A. So like this table we can explain where is the anticodon letter, what is the third end of the codon, how it can pair with the normal and unusual pairs. So you know very well, this is a flow of genetic information from DNA to RNA, RNA to protein. We are getting that central dogma of the biology. So next one is mutations explained based on the genetic code. First one we can go for silent mutations. Silent mutation